I am here today to talk to you on the genetic profile of hearing impairment in Africa. Hearing impairment can be classified based on the degree of hearing, and we are looking at childhood hearing impairment. So hearing impairment would be people that cannot hear below 25 decibel. We have two major factors that cause hearing impairment: genetic factors and environmental factors. The genetic factors, over 123 genes are associated with the condition, and these genes are well characterized and studied in the European and Asian populations. But in the African population, we are yet to identify the major causes of hearing impairment. I want to say that for us to give appropriate intervention to children with hearing impairment, we need to conduct research, identify the genes that are associated with the condition. Then this would help us to develop diagnostic tools for clinical testing of the genes. Then this would be incorporated into the newborn hearing screening program. We conducted literature on these various databases and identified 90 patients that were included for data extraction and analysis in this study. Majority of the studies were from North Africa. The numbers written in black are the total number of studies from the various countries, and the ones in red are the studies that's reported on consanguinity. A lot of the studies were also from North Africa, with only few studies from Saharan Africa. We have a lot of countries in gray, which means that we did not have publications from these countries on the genetics of hearing impairment, and they need to be investigated. A lot of genes have been reported to be associated with hearing impairment in Africa, making Africa no exception to the genetic heterogeneity of hearing impairment. The most genes studied on the continent is GJB2. And among the GJB2 variants, 4 of them were frequently or commonly reported. 3 of the commonly reported ones were from the North African countries, with only 1 from West Africa. A lot of the publications focused on targeted sequencing as the molecular method for investigating hearing impairment genes. This is very effective, but not comprehensive. So it would be very useful to use next generation sequencing approach to investigate hearing impairment in Africa. To conclude, North Africa is the most studied African population, with little study from sub-Saharan Africa. And the most common gene investigated is GJB2, with the 35-DELG as the most common variant reported, with only 1 variant that is common from West Africa, specifically Ghana. A lot of the studies use targeted sequencing. This is effective, but we think that the way forward is to use next generation sequencing approach and focus on familial studies. I want to use this opportunity to thank my supervisors and also thank the people who funded this work.